the Metropolitan Antiviral Drug Accelerator. As you can see on the next slide. So our mission, like essentially all of the AViD programs, is to develop new novel orally bioavailable drugs, small molecules to treat COVID-19, other coronaviruses, other emerging viral diseases with pandemic potential, all of it with a product profile that could be utilized in the outpatient setting. Now, the consortium, the core consortium, which is shown here in the middle, consisted of the Center for Discovery and Innovation, Rockefeller University, and I lead the program with Charlie Rice at Rockefeller. It involves other members of Rockefeller, as you'll see in a minute. But the initial consortium had Columbia, Aligos Therapeutics, Rutgers, and Memorial Sloan Kettering, with commercial partnerships from Merck and the Tri-Institutional Therapeutics Discovery Institute that is largely funded by Takeda. And has other partners as well. And then subsequently, we've added partnerships from Yale, Princeton, University of Arizona, and then UCSD. So the program, which you can see here, so these are the core project leaders. So we had 5 initial projects that two of which were focused on the main protease. That's work from Aligos, being led by Julian Simons. And they have work. In fact, one of their molecules will be going into phase 1 clinical trials at some point this year. And so it's moved very, very quickly. Program that I lead with Merck as a commercial partner is also focused on the main protease, and we have a number of advanced leads that hopefully will go into preclinical IND enabling and de-risking studies very soon. Tom Tuchel from Rockefeller has a number of targets that he's looked at, but in particular looking at viral proteases and methylase inhibitors for a variety of viruses, from corona, flaviviruses, and alpha viruses. Jingyu Zhu from Columbia is looking at modulation of RNA synthesis inhibitors, or the RNA polymerase inhibitors, and how, for example, nucleosides can be modulated to reduce some of the activity, and then trying to overcome this. And then finally, in the early phase, Alex Chavez, David Ho, and Yossi Sabo, initially from Columbia. Alex has since moved to UCSD. We're focused on approaches to develop small molecules that could be identified as pan protease or pan coronavirus inhibitors, but actually looking at a wide variety of viruses as well. And they have sort of a unique screening platform to be able to do this, and then to advance the work. Subsequent to the original application, we've brought on 4 developmental projects. One, Sean Brady from Rockefeller, who looks at uses a synthetic bioinformatic approach to develop natural products that have very potent antiviral activity. Alex Ploss, who's looking at inhibitors of yellow fever virus at Princeton. Brett Lindenbach, who's developing specialized tools for genomic editing of some of our prioritized viruses. He's at Yale. And then Liz Campbell, who's at Rockefeller, who is looking at inhibitors of RNA capping for a variety of coronaviruses and other viruses as well. We also have 3 mentored projects. one from Wamia Chowdhury, who's at Rockefeller working with Liz, targeting SARS-CoV-2 and the capping inhibitors. Shelley Katane, who's at Columbia, looking at the interactions of certain drugs in terms of their resistance profile, working with David Ho, and Subodh Samrat, who's at Arizona, looking at inhibitors of chikungunya virus, targeting predominantly the NSP2 protease. Now, to help these projects along, we have a senior leadership group, which involves myself, Charlie Rice, David Ho, and Steve Goff, to help move our programs forward. And just to address any operational issues, we have a very active scientific advisory board that's comprised of a number of industry professionals who have developed a number of antiviral drugs for hepatitis C, for HIV, for other viruses as well as drug discovery experts like George Rossano, who is an expert in PK/PD modeling, Arturo Casadevall, who has sort of a broad purview and is really the father of convalescent plasma therapy, but has a really strong view on overall drug discovery efforts. Susan Weiss, who's been working us very closely on MERS models, and you'll hear a bit about some of those interactions from Ching-Wen, but Susan, as a principal virologist, along with Richard Kuhn at Purdue, really help sort of provide some grounding for a lot of the programs and to advance it. And then finally, Joe Vaca, who had led medicinal chemistry at Merck for many, many years, works with a medicinal chemistry core to help advance much of the work that you'll hear about. So, this is where we're at. I mean, we essentially have gone from target to optimized leads. Many of the programs, in fact, entered into this program with either a lead or sort of a pre-optimized lead, for which the program was really here to be able to move these compounds forward. In some cases, the compounds started as active antivirals, like, for example, hepatitis C drugs, which had nice pharmacologic properties, but were not optimized against coronaviruses, in particular SARS-CoV-2. And so we've been able to move those forward and get them into the preclinical development phase en route to clinical phase development. And that is a key part of what we're about. We need to be able to develop move these molecules forward rapidly and get them into the clinic ultimately. Well, in order to do this, we've put together a series of pharma-style cores, and this program was really modeled after several drug accelerators, national drug accelerators that I've been running in the academic space, and in which we put in place these pharma-style cores for high throughput target screening. We have extensive virus screening. So we have a big virology core, which you're going to hear about, structure-based optimization. So in the early phase, this is really key to be able to go from a structure-based optimization through medicinal chemistry back to the virology, and to really begin to optimize this. At the same time, focusing in on the pharmacologic properties that would allow these compounds to be viable potentially in the outpatient setting, meaning that they have to have good bioavailability, stability, and be relatively non-toxic, or as non-toxic as possible, to be able to move forward. And then ultimately, they have to have really strong potency in our animal models. So we're constantly going around And at each phase, we have go and no-go criteria that we use to be able to advance potential compounds to the next phase to get to these optimized leads, which would then become preclinical drug candidates. So these are the core directors. Many of them actually have come from the pharmaceutical world who led cores at one time or another for pharma, whether it be high throughput screening, medicinal chemistry, and pharmacology, through our animal model cores, virology, and so forth, structural biology. And the key here is that the cores meet regularly. They talk to each other. The way we operate is that they support each of the projects to the extent that they need or want support. Data for each project is confidential within that project. It's never shared between projects, although within the cores, obviously, they're seeing lots of different projects and leads and chemistries. But we firewall it. It protects all the groups so that we can maintain the confidentiality and for intellectual property. And intellectual property is owned by each of the projects and universities or companies that are working with those groups. The MAVDA itself is really an umbrella group that makes sure that everything happens, but has no rights to any of the compounds that are being developed. So if you would like to hear a little bit more about the MAVDA program, how we work, we have a website, a newsletter, and feel free to reach out to us about specific projects. Now, I do want to mention that a key for the early stage development is optimization at the target, which is that we need direct antiviral activity. This program, we don't develop, as you know, we don't develop host-based drugs. We're developing direct antiviral compounds, which means that we have to be able to focus on the property of these compounds to inhibit virus replication. And so we've put in place an extensive series of cores, of which the virology core is front and center. It actually is a tripartite core that Ching-Wen will introduce to you, but it involves CDI, Rockefeller University, and Columbia University with different functionalities. And Ching-Wen can take you through how we've constructed that core and some of the specialized tools that that we've been developing to help move compounds along and get to our desired products. So I think with that, I'll just briefly introduce you to Ching-Wen. Let me stop sharing and I'll let him share. So Ching-Wen Chang was recruited to the MAVDA program from University of Massachusetts Medical School. Ching-Wen's had extensive experience working in virology. In his time at UMass, he'd looked at programs on both the effects of SARS-CoV-2 on the host and maternal health, but also was very much focused on drug discovery and trying to optimize and create tools that could be used to better screen compounds against SARS-CoV-2 and other coronaviruses. So with that, I will turn over the remainder part of the program to Ching-Wen, and he can take you through the virology program.
Okay, says Dr. Perlin, your kind introduction and comprehensive overview of MAVDA biological MAVDA project. So I just wondering, is my slide looking okay? Looking good?
Looks good.
Okay, thanks. And before I start my presentation, I would like to thank Jesse, Maggie, and Peter to coordinate this presentation to give us a chance to introduce our newly engineered cell model and how he can be applied to our current project to support our antiviral drug discovery project. So before diving into today's topic, I would like to take a few minutes to talk about the MAVDA biologicals. Overall, so we have three biological located at CDI, Rockefeller, and Columbia. And we allocate and share the resource across all biological to make sure all the MAVDA antiviral drug project can be fully supported. For example, under the supervision of Dr. David Perlin, so CDI biological has been focused on SARS-CoV-2 and the MERS antiviral drug development. So we have increased our capacity to the higher throughput screening. So for example, right now we can run around 150 to 200 drug testing per week. And most important is like we also, in past 1 year, we already identified the nanomolar scale lead compound, and those compounds have been moved forward to the preclinical study, in vivo study. And just like the CDI and Rockefeller not just focus on SARS-CoV-2, so they also focus on non-coronavirus target. So for example, they have like a very well-established cell assay for the flavivirus, alpha virus, and even the Zika, something like that. So in past year, so they have been identified the nanomolar scale, the lead compound, and they also is studying the mechanism of this compound and its relationship with drug resistance. So in the Rockefeller, so they have the bunch of like well-established cell assay for coronavirus and non-coronavirus. And they don't do like a high throughput screening. They just, because basically they test the pre-testing compound. So, and then to try to help our collaborator to identify the compound. And then I want to say, just like as one of our project in Columbia, we need to work very closely with MAVDA chemistry code for compound design or even for AI-based design. And under the supervision of Steve Gar and the Yossi Sabo, we developed two human cell line-based assay, which are A549 and the HU7 cell line-based assay, and combined with narrow luciferase reporter assay. So based on this model, we can quickly to check the compound and testing the compound activity against SARS-CoV-2 infection. So basically, we just need to like 2 days, so we can have like IC50 and the CC50 data back to our collaborator. So in that case, can help them to develop and design a more compound for the future for the further testing. And because we also use like a plasmid-based like a reporter assay, so we also have the ability to study and develop an assay for drug resistant research. Overall, I just want to say our all three biological are well developed and are working very well. So far we have testing over like 1,000 compound and identified very promising compound, and some of them already move on the preclinical in vivo study. And this table just give you an idea like what type of virus strain are available in our biological, so like coronavirus and non-coronavirus. And because each biological currently focus on the M-Pro and P-Pro co-proteins of coronavirus. So we also have several luciferase-based reporter strain in each biological. So for example, we have a Washington strain from UNC. We also have another Washington strain, which is plasmid-based. And we also just received like a MERS nano-luciferase from the UMC as well. And we also working on the MPA. So in the near future, we're gonna have the Delta and Omicron BA1 nano-luciferase reporter virus strain. So we are going to use those reporter virus and integrate into our current high throughput screening workflow. And this slide just give you an idea how our workflow look like that at CDI biological. So basically, we have a capacity for 96 well and the 384 well base assay, because we have a very nice equipment to do like a cell dispensing and compound dispensing. Even we have a Hamilton, so we can run automatically virus infection in our PSO3 facility. And then here, so we basically use like image-based, fluorescence-based readout, and also like a narrow lucifer space readout to identify compound IC50 data and also the CC50 data. So each state we have a SOP, standard operating procedure, to make sure we can generate high quality of data, and then to generate those accurate IC and IC50 and the CC50 data back to our collaborator. Rockefeller, so basically they running a pretty low throughput screening because those compounds, like I say, had been testing. So, but they have like a well-established cell assay, so they can also easily based on the image base. For example, they have image Express and citation 7 and operator to run like an image-based assay to help to determine like compounds IC50 data. And they also have ability to study the compound and his mechanism and how to involve in to generate virus resistance. So they have been identified several promising compounds and also design the assay to study the mechanism of virus resistance. So that's ongoing. And for the Columbia Biological, so here, because our collaborator, they usually need us to run the pre-assay. So here we designed 2 human cell line based assay. The first one is A549, which express ACE2 and TMPRSS2. And the second one is using the cell line Hu7 overexpress ACE2. Both of them are stable cell line. So basically, based on the narrow luciferase reporter strength, so we can easily to get the IC50 data and CC50 data within the 2 days, and then get the data back to our collaborator, and then they can design more the compound for the further test. And we also have a capacity for the seasonal coronavirus. Here we use MRC5 based cell assay, so both assays are available. So far our capacity in Columbia, we can easily to run 25 to 50 compounds per week. And then we can generate the data in 4 days. So as you can see, each biological currently focus on the human coronavirus. And we know like MERS, MERS virus and also SARS-CoV-2, both of them are the lethal coronavirus. And so in this idea, because when we test the MERS COVID-2 and the compound against MERS COVID-2, we want to understand their activity to against MERS or either SARS-CoV-2. So in muscle case, we have tested the same compound in the 2 different cell lines. Sometimes it's not easy to make a comparison because basically this data generated for 2 different cell lines. So based on this idea, we are thinking whether we can create a human cell-based cell line, and then this cell line can be applied for SARS-CoV-2 and the MERS infection. So based on this idea, we're just thinking about probably we can transduce the 3 viral receptor like ACE2, TPP4, and TMPRSS2 into the same cell line. Because once we have this cell line, so cell model, so can very helpful to support our current antiviral drug discovery. And we also can use this cell model to study the virus pathogenesis. So based on this idea, so we just look at the current the most commonly used cell line like A549 base, or like a Karu cell, Keiko cell, and Hu 7.5 cell line. And because here, as we know, the lung tissue is a major target of coronavirus. And so here we just focus on the cell line derived from the human lung tissue. And the other thing, the Karu cell is also a good cell model, but the drawback is this cell line going very, very slow. So it's not a good model for the high throughput screening. So here we just look at and focus on the A549 based cell model. And we also thinking about what kind of criteria we have to think about that to create this ideal cell model. So the first one is a cell growth rate, because to do like the high throughput screening, so we want the cell have a normal growth rate. for example, like a carousel growing very, very slow. So it's not an ideal model for high throughput screening. And here we compare to A549 ACE2 Tempest cell line. So the first one is I made from the UMass for the ACE2 Plus. And the other one is commercial available cell line from the InVivoGen. So both of cell line are A549 based and also co-express ACE2 and Tempest 2. But as you can see here, compared to the one from the InVivoGen, so this cell growing a little bit slower than ACE2 Plus cell. So in this case, we probably consider just use ACE2 Plus for the multiplication to create available cell model for our study. So the second one, criteria I check like a viral receptor expression. So here, as you can see by the qPCR, I checked ACE2, which is highly expressed in A549 based cell model compared to like a carousel. And I also check a Tempest 2. So both SARS-CoV-2 viral receptor are highly expressed in both cell line. And the other concern is that, well, we prefer to have like a homogeneous cell model. So in this case, we can have a more consistent data and even have like a more stable infectivity across like a different patient number from early to late patient number. So in this case, for example, as you can see here in this image data, so ACE2 and Tempest receptor are ubiquitous expressed in each cell receptor. And then we also want to think about is the ideal cell model. So it's able to tell the potency difference between the different compounds. So here, just give an example. So we use the Remdesivir and AViDD 1931. So both of them are targeting viral replication. But as you can see here, based on the IF image result, so the Remdesivir showed a better potency than AViDD in this case. And then we also test like whether this cell model, we also want to consider whether this model can show like a stable infectivity from like earliest patient number through like a late patient number, like a patient number 20. So as you can see the data here from like a patient number 9 through the patient number 20. So they show a very similar comparable infectivity. And then the last criteria we are thinking about is whether this cell model can really fully support the viral replication inside the cell. So here, just like an EM image give you an idea. So you can see this data, an image generated from ACE2 Plus cell. So you can see here like Omicron BA1, his viral protein are very actively replicating on the rough ER. And if you look at the mitochondrial structure, you also can see once virus infect to the cell, so make the mitochondria look pretty sick. So in this case, we can image mitochondrial function probably have some impact, for example, like ATP level. And then the other part is, on the other hand, we want to test whether this cell model can be applied for the study of host viral cell interaction. So here we transduce like a spike plasmid into ACE2 Plus cell. So, and then also run the qPCR to check the interferon induced transmembrane protein expression. So as you can see here, once we transduce delta spike or even the Omicron spike here, but as you can see, a delta spike has could be a very strong inducer. So once expression the spike protein inside the cell, so those interferon relative gene can be induced. And those data are generated from the A549 ACE2 Plus. So based on those criteria, so we decide to based on the ACE2 Plus cell model, and then transduce another viral receptor DPP4 for the MERS entry. And I am not going to talk about the detail here because we play around a lot between about the ratio between those three receptors. So and overall, eventually we got the couple the different cell models. So I'm gonna just show you the data here. And but I just want to let you know this newly engineered cell model, they co-express the three receptors, SE2, DPP4, and Tempest 2. So here, just I give you an example how we test the new engineered A549 cell model. So here I use the image-based assay. So as you can see here, so we're seeding the cell in 96 well, and next day we're gonna treating cell with like a reference drug like Metropolitan, and also challenge with the virus. Here I use the MERS virus from the 2012. And then 48 hours later, here is 40 hours later, not 24, sorry. So we fix the cell and scan the viral specific protein like a nucleic capsid, and then use our high content image reader to scan the signal from each well. So as you can see here, so the red color stands for the MERS nucleic capsid signal. That means those are infected cells. And that means like a total cell number here. So you can see the dose-dependent effect here. So when we increase the neometric concentration, we can see the infectivity going down in this case. And so we also use the same similar strategy to test like a SARS-CoV-2 Omicron variant like SBB 1.5 and BA5. So we represent those data as like a heat map. So it's more easier to see. As you can see, if we just test using the reference drug like a Metropolitan, so look like Metropolitan showed a better potency against SARS-CoV-2 infection than the MERS here. And then the next step we want to like, because as we know, ACD part cell 9 can show very strong virus-induced cytopathic effect during the virus infection. So in this case, we also want to like use like CP-based assay to determine compound EC50 data. So I just give you the idea here. So this is the image I took from a post-infection 24 hours, and this is 48 hours, and with the high to low MOI. So as you can see here, the CPE is very easy to observe post-infection 24 hours. And this CPE effect can be rescued by treating cell with the neometropoir. So based on this idea, so we also optimize the condition. So we test two different like condition, for example, condition 1 and 2 here. So in condition 1, as you can see, we have a higher signal to noise ratio. But in the condition 2, we have a lower signal to the noise ratio, probably just 3. In this case, we think about the condition 1 might be the better for our CP-based assay to determine compound EC50 data. And we also like test the same idea and apply to the Omicron variant. So for example, EG 5.1 and SBB 1.19 and 1.16. So we also can easily observe the virus-induced CPE effect here. And we also test this cell model challenge with like coronavirus like 229E here. So we also can see the very, very clear CPE effect. So in this case, I would say based on this data, so it's possible to develop the assay and based on the CPE to determine the compound EC50 data. And I forgot to mention here, so we use like stereotyped growth to determine the CPE effect. So for example, when the virus induced CPE, and then in this case, gonna cell dying. So the cell dying, so the intracellular ATP level is going down. So in this case, we just need to use stereotyped growth kit, which is narrow to surface space assay to determine the CPE, the percentage. So in this case, we can have an idea about the compound EC50 data. And to confirm our CPE-based data, so we also apply the immunofluorescence-based assay to confirm this model is highly permission to all SARS-CoV-2 variant. For example, here we test the EG 5.1, SBB 1.9, so the signal is very clear. And the most important is we also test like and challenging those cell model with MERS virus. And here we test 3 different models. So as you can see here, the model 1 show the very nice infectivity, even the model 2. But for the model 3, we at the same condition. So look like this cell model is very sensitive to virus infection, because once MERS infect to the cell, it immediately also induce cell dying. So this is something we don't want to see, so I can explain later. Yes, and so we also, based on this engineering cell model, we challenge the cell with MERS and also SARS-CoV-2, because we want to understand what's their replication kinetic data look like that. So here is we fix the cell at 24 hours, and this is 48 hours. So as you can see here, using the same MOI, the same cell line, so you can look at like a top panel. So once the cell challenge with the MERS, you can see the MERS, his replication kinetic is faster than the SARS-CoV-2, because at the 24-hour time point, so we can barely see like SARS-CoV-2 positive cell. But for the 40 hours, we can easily observe like over 50% cell are nucleic capsid positive. So based on this idea, so we know this model also can be applied for study the pathogenesis between the two different virus strains. And then we go back to like to talk about the model 3, because based on this data, we see even use pretty low MOI, like a 0.0001, just 24 hours muscle cell killed by the virus. So we just get closer to look at like this data. So it's very clear that so those cells are killed by the spike-induced the cell fusion and spike-induced CPE. As you can see here, this is 24 hours. Use a pretty low MOI of 0.01, and then 40 hours muscle cell die. That's because cell fusion. But if you look at like the same condition from the model 1, so just 24 hours, even use a little bit higher MOI, like MOI 0.1. So the virus still can have very efficiently replicating the cell, and the cells still look okay. They are not really dying. So to scope like infectivity and also the total cell number, so we use citation 10 to quantify the nucleic capsid positive cell across the 3 different models, from the model 1, 2, 3. So overall, the infectivity look pretty good, just over like 40%. But if you look at like the DAPI signal, I mean, represent for the total cell number in the model 3, so even use pretty low MI. So compared to the other model, so almost half of cells are dying because of spike-induced cell fusion. So in this case, we think about probably the model 1 or model 2 should be better for like our antiviral drug screening and also for pathogenesis study. So here just give you like a model to explain so why like, because basically when the spike-induced cell fusion, so they're gonna kill like a cell like eventually. So in this case, the key to make ideal model for the drug screening and also for the virus pathogenesis study is the ratio of those receptor. So here just give you an idea in the model 1, so we manipulate the expression level of those 3 receptors. So in this case, you can see even in MOI 0.1 and 24 hours, cells still look okay. They are not really dying. And for the model 3, so because we know in this cell model, they express the pretty high level of those 3 virus receptors. So this makes this cell model really very sensitive for virus infection. So that's why just use pretty low MOI at post-infection, you can easily to see the cell dying because of virus and the spike-induced CPE. So we also, based on this, so we also like to test the IC50 compared to both cell model. So here is I use the Metropolitan as reference drug, so to determine the IC50 by the image-based assay. So as you can see here, so we expect to observe the higher IC50, as you can see here, compared to the model 3. Because in the model 3, as we know, once cell start undergoing cell fusion, wherever the virus is mature or virus particle is mature or not mature, so the cell they just die. So in this case, I would say because in the model 1 is able to generate higher amount of the virus particles. So that's why even the same condition, we can see the IC50 data is higher, a little bit higher than the one in Model 3. To confirm this, because in Model 3, we know probably image-based is not a good assay to determine the IC50, because we know post-virus invasion, most of cell, 50% cell dying. So here we use a CPE base, so to confirm this data. So, and then we got, as you can see, the blue curve here. So we got a similar like IC50 data, very close to the 100. Yeah. And so based on this data, we're thinking about probably Model 1 it's a better model than the Model 3. So we decided to follow up and focus on the Model 1. So use the Model 1. So we also like test like Omicron variant, like for example, SPD 1.16 and 1.19 here. So as you can see, the EC50 data is very close. But compared to like the MERS, so oh, sorry, I don't have data to show here, but as we know, like if use the same assay to look at like the data from the MERS infection, so we know the EC50 data is higher, absolutely. So so I summarized the data, make a table here. So based on this model, we test with MERS, SARS-CoV-2, and recently Omicron variant, even the 229 and OC43. So we can easily observe the virus induced CPE, and this CPE can be rescued by treating cell with nirmatrelvir. So based on those data, so we think like another question we want to ask is whether this model has potential and can be applied for high throughput screening. So like I said before, because each our step we have SOP for our high throughput screening. So for the step 1, we have a well-established SOP. So even if, for example, if today our customer want us to test like to run the pre-test to test like 100 compound and get a database to them in 1 week. So we already identified protocol. So even we can use like a frozen cell from the freezer, and after plating the cell, just take 3 days. So we can have like over 10 million cell in the large T flask and over 90% cell viability. So in this case, just take 3 days to get enough cell number for the seeding, and also just for the compound dependency. So then we can use Hamilton to do automatic virus invasion. And here just give you an idea. So it's just 1 minute like video, so you can see how the Hamilton works. And just want to mention this video was taken by our senior research associate Jerry. Yeah, so he used like Hamilton to do automatically virus invasion. So in this case, you don't have to do any kind of invasion manually. So I think based on this workflow, I think is very useful if, for example, you want to run like the library screening. So in this case, we don't have to do it manually. So in this case, we're gonna have a more consistent data. And so based on this workflow, so we just use the small size of library, which include like 116 library. So to test our workflow. So here we use the 384 well format. So if we run the 4 replicate, so each play we can test around the 80 compound in the single play with a single dose. So if we run the 3 replicate, we can run around like 106 compound in each play. And because we have like a small size library, just have 160. So in this case, we just need to prepare 4 play, 2 play for the compound IC50, another 2 play for the CC50. And here we use like SARS-CoV-2 reporter strain, which carry like a narrow luciferase. So that means post the invasion 24 hours, we can have this data. And so here I use like remdesivir as an internal control. So as you can see here, like a red dot. So remdesivir can significantly suppress virus activity in this case. And we also use like a 2 IC as the cutoff threshold. So in this case, we identify 2 potential the hit compound here. So, and then in this case, we just use the dose. We're going to follow up by running the dose response curve to validate and identify this compound potency and efficacy. So overall, based on this idea, so I think our newly engineered cell model, I think is look promising. And so we also gonna integrate this new cell model to our current high throughput screening protocol. So I just give you the last slide. I want to give you like the summary and our next step. So based on those data, I believe our newly engineered A549 cell model are highly permission to the MERS and SARS-CoV-2 infection, including emerging Omicron variant like EG5.1, PQ1, and SBB series. And this engineered A549 cell model also is able to show significant cytopathic effect post the virus infection. And this effect can be rescued by adding like a protease inhibitor like a MAVDA. So in this case, it's more easier to, to determine the compound, the ECP data, not just based on the image base. And the novel A549 engineering cell model also show this potential for antiviral drug discovery. For example, like can be integrated into our high throughput screening assay. So we just received like a MERS luciferase reporter virus trend. So in the future, we're gonna use like SARS-CoV-2 luciferase and the MERS luciferase trend and also this new cell model to integrate into our current high throughput screening. And supposedly it should be easier to identify the key compound and then move on the next step. And we also, based on our like the different time point assay, 24 hours and 48 hours, look like the MERS virus showing the faster virus replication kinetic than SARS-CoV-2. And so to confirm this, we are running the qPCR and running prior assay to confirm this data to make sure this model can be used for studying both distal coronavirus. And so still ongoing, but we also still characterize this novel A549 cell model. So if you are interested, so feel free to contact our project coordinator. So I will introduce like later. But based on those data, I think this new engineered cell model look promising and can be applied for SARS-CoV-2 and the MERS virus infection as well. So so here I want to like test this opportunity to thank our like the team member then to help me to test like this new engineered cell model and give their feedback to me. That's very helpful. And I also want to like thank our project leader, Dr. David Perlin, and also Charles Rice, and also our co-director, Steve Goff and Jose Sabo. So they are very supportive to help me, very supportive so I can just explore this idea and carry out this idea to establish this cell line. And also want to thank our scientific advisory board member, Susan Weiss, and her manager. So they provide very useful and valuable suggestions to make me have a more idea to create this cell model. And I also want to say thanks like Sol and also Alex to provide the TPP4 full sequence and also the plasmid. And last but not least, so I also want to thank our like CDI facility director, Steve Parr. So he has been very supportive and then to find a resource for me to create this cell line. And also the Madhu, our project coordinator, so to help me to contain like Anthony to step and coordinate the meeting. So in this, so we already submit the IP application, so that's very helpful. So I'm gonna just stop here. So and I will be happy to take any question from you.